Good evening, plant physiology students. Welcome to week 12 of semester. And we're really at the busy end of semester. Everyone's busy finishing off assignments and we're busy marking. This is our last class, our last online class for plant physiology. And this lecture tonight, we'll be looking at plant growth and development. So let's make a start. Oh, hello, can you hear me okay? Oh, terrific. All right, let's start here with an introduction to growth and development. So we can start by thinking about animals, and of course, unlike animals, plants have a very sedentary lifestyle. So plants have never evolved that complex anatomy that enables mobility in animals. Instead, we find in plants a very rigid anatomy, which is adapted, of course, to the capture of light energy and the uptake of nutrients. So as a consequence of this, plant cells tend to be firmly attached to other cells in a very inflexible type of matrix, and this imposes a lot of constraints on how the plant grows. So cells are added progressively to the plant body through the activity of meristems, which you're all familiar with. By contrast, in animal development, particularly in the development of the embryo, and this includes the formation of those three primary cell layers in animal embryos, that type of development is characterised by migration of cells to new locations. So you might remember last year we talked about the movement of those neural crest cells throughout the embryo to give rise to different tissues, including tissues of the nervous system. So this lack of mobility for plant cells means that plants have to adapt to their local environment, which occurs on both a physiological level, but also through very flexible patterns of development, if you like, that characterise that vegetative growth, and through regulated proliferation and differentiation of cells from meristems. And in this way, plants can produce a variety of complex forms which are very adapted to their local environment. Oh, that's okay. Then it's good that you're here tonight, if you haven't watched this lecture. Okay, so let's look at this image here. This illustrates the three major phases of the seed plant sporophyte development. And remember that the sporophyte, this term or this word here, sporophyte, refers to the dominant part of the organism's life cycle. And in the case of flowering plants, the dominant part of the life cycle, the sporophyte, is the diploid part of the plant's life cycle. In other words, what we can see here. So the three stages here are embryogenesis, vegetative development, and reproductive development. So that's pretty straightforward. So we start with embryogenesis here to the left, and this describes the process where a single cell, this zygote here, divides to transform into a multicellular organism. And at first, this just has the characteristics of just the very basic level of organisation. And of course, this takes place mostly within the ovule found in the carpel of the flower. Then vegetative development begins with germination. So once the embryo breaks its dormant state and it mobilises all those stored reserves, so for example, the starch reserves in the seed, and then it commences a period of vegetative growth. Depending on the species, germination occurs in response to different factors, including, of course, moisture, cold, heat, or light, and this depends on the embryo. And at first, it survives on its reserves in its cotyledons, or it might be surviving on its reserves in the endosperm of the seed, and that characterises the grasses. Then the seedling builds on its form through the activity of its shoot apical meristem and the root apical meristem. And then further development enables the seedling to become photosynthetically active. So this enables further vegetative development. And it's interesting that unlike animals which exhibit determinate growth, that is, they grow for a certain period and then they stop at a certain point. But vegetative growth, which we see in plants, is said to be indeterminate. That is, it's not predetermined and it's subject to variation with no definite endpoint. Then the third stage of development is reproductive development, which occurs after a period of vegetative growth in response to both internal and external cues, such as size, temperature, or maybe even photoperiod, that is day length. And this transition from vegetative to reproductive development, it of course involves the development of specialised floral meristems that give rise to flowers, as we see here. So this is all summarised on the next slide. Here we go. 3 major stages of sporophyte development. Okay, now we're going to look at some of the fundamentals of plant development, starting with embryogenesis. In seed plants, of course, embryogenesis transforms that single-celled zygote into the more complex embryo found in the mature seed. And this process establishes the basic architecture of the plant, and it includes developmental processes such as morphogenesis, that is the elaboration of form, organogenesis, so the formation of functional structures, and histogenesis, which is the differentiation of cells to produce various tissues. Now, a major feature of these processes is the development of the apical meristems at the tip of the shoot and the root axes, which are the keys to sustaining that indeterminate pattern of vegetative growth. And finally, the embryo undergoes complex changes that enable it to withstand long periods of inactivity, that is dormancy, and also changes which enable it to recognise and respond to environmental cues that signal that embryo to resume growth, that is germination. Oh, terrific. We've got someone else online. Good on you, Lachlan. I thought after today's prac workshop in wine chem, none of the students would be here. Okay, we're going to examine embryogenesis in a dicot. And this particular dicot you've been introduced to. This is the model plant called Arabidopsis thaliana. It's a little weed, of course, not really useful for anything else, but it's been extensively studied by scientists around the world. Its genome was the first plant genome to be sequenced. It's very well sequenced, very well annotated. It's a very small, fast-growing plant, so it's really been the ideal model plant for many, many years. All right, in Arabidopsis, there are 5 stages of embryogenesis, starting with stage number 1, the zygotic stage. So this is the first part of the diploid life cycle, that is the beginning of the sporophyte stage. And this arises as a result of fusion of the haploid egg cell with the haploid sperm cell to form that single-celled zygote. And then what happens is that there's polarized growth of that cell, and that's followed by an asymmetrical cell division event to give rise to a small apical cell and a basal cell. And we'll look at that in just a minute. Stage number 2 of embryogenesis is the globular stage. And here, one of those two cells, the apical cell, undergoes a series of cell division events by mitosis to generate a spherical 8-cell globular embryo, which of course exhibits radial symmetry. Then additional cell divisions form additional cells to create an outer layer called the protoderm. And this later on becomes the outer epidermis. Next, we have the heart stage of embryogenesis. So rapid cell division in two specific regions of the embryo on either side of what will later become the shoot apical meristem will form the primordia to the cotyledons. So this will actually give the embryo bilateral symmetry. Okay, stage number 4 is called the torpedo stage. Here we see cell elongation throughout the embryo and further development of the cotyledons occurs. And then finally we have the mature stage. So here the embryo and the surrounding protective seed lose water. The embryo becomes metabolically inactive as it enters dormancy. And storage compounds will accumulate in the cells at this stage. And here it is, the stages of embryogenesis. So in A, we can see the zygotic stage. So that original zygote has divided just once to form these two cells, the apical cell and the basal cell. Then in the next probably 3 images here, we can see various stages of development which are referred to as the globular stage. And here this outer cell layer called the protoderm has formed. And as I said, that will give rise to the outer epidermis. Down the bottom here, we have an early heart stage and late heart stage, and you can see why they're referred to as heart stage. At this early heart stage, we can see these two regions here, which are going to give rise to the cotyledons. So they're referred to as the cotyledon primordia. Now the cotyledons are forming, as you can see. Next stage here, this is called the torpedo stage. And now the shoot apical meristem starts to form right here. And then finally, we have the mature stage in this final image. Oh, sorry, back to the torpedo stage. Down here, this will become the root apex. Oh, you need a refresher. Yes, so do I. Don't worry. Absolutely. Okay, so a comparison of embryogenesis between Arabidopsis, which is a dicot, of course, with a monocot such as rice, illustrates some differences in how it all unfolds, but a characteristic feature of both can be generalised to all seed plants, really, and this relates to the establishment of polarity. This polarity commences in the zygote, and the embryo then becomes increasingly polarised throughout development along 2 axes. The apical basal axis, which runs between the tips of the embryonic shoot and the root, and then also a radial axis. which is perpendicular to the apical basal axis. That is, it extends from the centre of the plant outwards. Now, an early sign of this apical basal polarity is even seen in the zygote itself. The zygote elongates about threefold, and then it becomes polarised with respect to the composition within the actual cell itself. The apical end of the zygote becomes densely cytoplasmic, and that's in contrast to the other end, the basal end, which contains a very large vacuole. And these differences are captured even further when the zygotic cell actually divides, and it divides asymmetrically to produce a short, cytoplasmically dense apical cell and a longer vacuolated basal cell. And hopefully we'll see that on the next slide. And we do. So let's start with the embryo sac on the left. You might remember an image like this from last year. And of course, this is where we find the egg cell. And once that egg cell has been fertilised, of course, the single-celled zygote is formed here. But let's look at the zygote here in the middle, enlarged somewhat, and the zygote becomes polarised, as we said on the previous slide, and it's polarised here as pictured with the formation of this large vacuole at this end of the zygotic cell, and then just dense cytoplasm at the other end of the zygotic cell. All right, then this cell divides asymmetrically, so about here, not right in the centre, and that produces two cells that we can see here, the smaller apical cell in green and the larger basal cell. And interesting to note here that nearly the entire embryo and ultimately the adult plant will actually derive from this apical cell and a small portion of the basal cell itself. Most of this basal cell that you can see here will actually form what's called a suspensor, which attaches that embryo to the vascular system of the parent plant. Okay, so let's revise. Nearly the entire embryo, therefore the mature plant derives from the apical cell. The apical cell will divide to generate an 8-cell globular embryo, and the basal cell, or rather most of that lower basal cell, will divide to produce the suspensor, which attaches the embryo to the vascular system of the plant. So as we said, only a small portion of this basal cell, which is called the hypophysis, will become part of the mature embryo, and it actually forms part of the root apical meristem. All right, and here it is, illustrated. So here the apical cell, so this region up here, has divided to produce an 8-celled embryo. Only we can't see the 8 cells, we can only see 4 there. And 2 domains are labelled here, the apical domain and the central domain. Here the basal cell has also divided, and we can see this upper region in blue called the hypophysis, and the lower region which is going to form the suspensor. Okay, here we have the 16 cell embryo, and then the early globular embryo. So by this early globular stage, broad areas of cells can actually start to be related to cell fate. So the apical region, up here in green, the apical region will give rise to the cotyledons and the shoot apical meristem. This middle region here will give rise to the hypocotyl, that is the embryonic stem, and also most of the root and part of the root apical meristem. And then, of course, the hypophysis, which is derived from the uppermost part of the suspensor, will give rise to the rest of the root meristem. Okay, and that's all summarised here for you. So here we can see how the embryo progresses from that earlier, from the early globular stage through to the late globular stage. I'm not sure about this transition here, but maybe transition to the heart stage, early heart stage, late heart stage, which we can see here, formation of the cotyledons occurring here. And here we have the seedling, and it's been nicely colour-coded so that we can actually see which parts of the seedling are derived from which cells in the embryo itself. So most of the cotyledon cells, shown here in dark green, derive from these cells up here, and so on. All right, auxin. Everyone's familiar with auxin, and it pops up all the time, even here. So during animal development, substances called morphogens play very important roles in providing what are referred to as positional cues to cells during embryo development. That is, they form concentration-dependent gradients in the embryo, and these gradients are responsible for determining where certain cell types develop, where certain tissues, limbs, and organs and so on develop. Now, it's thought that auxin in plants may act as a morphogen in plant embryogenesis, and that's because they've found that auxin or any of its synthetic analogues can be used to induce the formation of embryos from plant somatic cells, that is, non-reproductive plant cells. Okay, evidence of a role for auxin in embryogenesis. So late stages of embryogenesis have also been shown to be sensitive to auxin levels, as demonstrated here. So let's look at what we've got here. Here in B, we have a wild type or normal Arabidopsis embryo. And to the right, here in C, this is a mutant embryo. And it's caused by disrupting a gene called the PIN1 gene, which is actually responsible for auxin transport between plant cells. And you can see that obviously it's had quite an effect here on morphology. And here's another mutant embryo. This is an embryo of Brassica juncea, and this embryo was incubated in trans-cinnamic acid for 10 days. This chemical is an inhibitor of auxin. So all of this suggests that auxin is essential for normal embryogenesis. Okay, so they've used lots of different techniques, including mass spectroscopy, immunodetection, reporter genes, protein localization, studies, and so on. And using all these techniques, they've actually developed a hypothetical map which shows the predicted movement of auxin during development of the embryo. So here we see at the 2-cell stage a maximum concentration of auxin in the apical cell. Then at the globular embryo stage, we can see auxin. Maximum concentration of auxin accumulates in the hypophysis. And then even at the early heart stage, auxin's transported according to the direction of the arrows that you can see here. And high levels of auxin are maintained in the cells here, that is the cells that are going to give rise to the cotyledons, and also in the cells of the hypophysis. So this suggests that auxin gradients determine apical basal polarity of the developing embryo. All right, let's move on now to meristematic tissues. And a meristem can be defined simply as a group of cells that retains the capacity to proliferate by mitosis and whose ultimate fate remains undetermined. And the two major meristems, of course, are the root apical meristem RAM and the shoot apical meristem SAM. Both of these meristems features a defined cluster of cells called initials, and these are characterised by their slow rate of division and undetermined fate. Oh, don't you love skipping all these slides? Okay, so let's start with the basic features of root development by distinguishing zones within the root with distinct cell behaviours. So firstly, we have the root cap, which occupies the lowest part of the root. It contains a very unique set of initials which are displaced away from the meristematic zone in the actual root apical meristem. And these are cells that cover and protect the apical meristem from injury as the root tip pushes through the soil. Root cap cells also function in the perception of gravity, which we mentioned briefly as gravitropism during the prac workshop. Root cap cells also secrete certain compounds that help the root penetrate the soil and to mobilise mineral nutrients. Okay, the second developmental zone of the root, here we have the meristematic zone, which lies just under the root cap. Here we find the root apical meristem initials, and these divide to produce cells that will divide further to differentiate into the various tissues that make up the root. Okay, developmental zone number 3, the elongation zone. This is the site of rapid and very extensive cell elongation, although some of these cells actually continue to divide while they elongate within this zone. And even though this is still happening, the rate of division will decrease eventually to 0 the further away that these cells get from the actual meristem. And finally, we have the maturation zone. This is the region in which the cells acquire final differentiated characteristics. And here, cell division and elongation has ceased, and this is where lateral organs such as root hairs and lateral roots will form. Okay, illustrated here is a primary root, and you might be slightly familiar with this image because I use the same image in living systems, in one of the living systems lectures. All right, but let's revise. So primary root showing the root cap down here, the meristematic zone, so this region here, which also includes this little region in yellow called the quiescent centre. And of course, further back, we have the elongation zone, and then further up, the maturation zone. So in root tips of Arabidopsis, these zones that we're looking at here occupy only about 1 to 2 millimetres of the actual root tip. So let's work our way up the root here and look at actually what's happening. All right, root cap cells, root cap initials would be along here somewhere. Then we have this region called the quiescent centre. And then here we have the maximum rate of cell division in the root apical meristem, or part of it. And then cell division ceases in most layers around this region. And then we can see some differentiation of tissues starting to occur. So in green, we can see the first sieve tube element of the phloem starting to differentiate here. This region, maximum rate of cell elongation is occurring. A little bit further back, the first vessel elements begin to differentiate. So the xylem is starting to form here. Endodermal cells in brown will start to differentiate around this region. Further back, we have mature vessel elements. So now the vascular system is completely formed, and those surrounding cell layers, the periderm, the endodermis, and so on. In this maturation zone, of course, this is where the root hairs will start to form. A little bit further back, we can see a very young developing lateral root primordium, and here's one that's emerging. And now we have all the cell layers being formed, completely formed and differentiated. Epidermis, the cortex, pericycle, endodermis, and of course all the vascular tissue has been formed. Okay, so the origin of the root cells and tissues can actually be traced back to specific initials located in the root apical meristem. At the centre of this region is the quiescent centre, QC, and it's named for its low rate of cell division compared to those surrounding initials of the root apical meristem. There's only about 4 to 7 of these cells in the quiescent centre of Arabidopsis. Now, adjacent to the quiescent centre, there are 4 distinct sets of initials, and each of these sets can be defined in terms of their position and the cells and tissues that they're going to produce. So these 4 sets of initials are the columella initials, the epidermal lateral root cap initials, the cortical endodermal initials, and the steal initials. So let's look at these here. So the columella initials will divide to form the central portion of the root cap cells. Epidermal lateral root initials will form the lateral root cap cells and epidermis. Cortical endodermal initials form the cortex cells and endodermis. And steal initials will form the vascular system and the pericycle cells. Okay, and here we have it all nicely illustrated. So in A, we have an actual longitudinal section through a root tip, and it's showing outlined in green. This is the root apical meristem region. And this region contains all those initials that will give rise to all the tissues in the root. Okay, the same thing is shown here, but the individual initials have been obviously enlarged and coloured. So we've got columella initials here in light green, and these will form the central portion of the root cap cells. That is, they'll form these cells of the root cap here. Then we've got the epidermal lateral root initials, so on this side and on this side as well, and these will divide to form the lateral root cap cells, and also the epidermis shown here, a couple of epidermal cells there and an epidermal cell just there. So you can see the different groups of initials that will give rise to all those different tissue types. And welcome auxin once again. Auxin appears to play an important role in positioning the root apical meristem and guiding its behaviour. The position of the quiescent centre coincides with a maximum concentration of auxin, and models have shown that auxin moves down from the shoot through the central regions of the seedling and concentrates in the quiescent centre. The auxin is then transported outwards via PIN protein transporters, and then auxin moves upwards towards the shoot. And that should be illustrated here on this slide. So we can look at B, this model of auxin flow here, which corresponds to the root tip that we can see on the right-hand side. So firstly, just following these arrows in the centre here, auxin's moving down from the shoot through the centre of the root tip as per these arrows, and then it concentrates here in the quiescent centre of the root tip. And that's also shown here by what's called a reporter gene, GFP reporter gene in green. Okay, from there it starts to move laterally. We just follow the arrows, and then upwards towards the shoot through the outer cell layers. So once again, auxin's involved. Okay, studies have shown that another hormone called cytokinin, which we didn't look at in the prac class, acts in opposition to auxin. So we saw during the prac workshop that auxin is synthesised primarily in the shoot apical meristem and transported basipetally towards the roots. In contrast, cytokinin is synthesised in the root and is normally transported acropetally. In a similar opposing fashion, auxin promotes root development, both in tissue culture and in vivo in plants, whereas cytokinin has an opposite effect. It suppresses root development and promotes shoot development. And studies have shown that cytokinin signalling begins very early in embryonic development in the hypophysis of the globular embryo. And when the hypophysis divides, cytokinin expression is lost in the basal cell, but it's retained in the apical cell, which then divides even further to form the quiescent centre. Okay, so just like the root apical meristem, the shoot apical meristem, pictured here, has to maintain sets of undetermined cells that enable that indeterminate growth. And just like the diversity that they see in the size and the structure of root apical meristems from different plant species, shoot apical meristems also vary from one species to another. So, for example, a nice example is in your textbook, your prescribed textbook. The largest shoot apical meristem found in vascular plants is found in the cycads, and they measure over 3 millimetres in diameter. At the other end of the spectrum, there's Arabidopsis, which has a shoot apical meristem of only about 50 microns in diameter and contains only a few dozen cells. Okay, the shoot apical meristem also has distinct zones and cell layers, just like the root apical meristem. At the centre of an active shoot apical meristem is the central zone, CZ, which contains a cluster of infrequently dividing cells, which can be compared to the cells of the quiescent centre in the roots. Flanking region is called the peripheral zone, These divide more frequently to produce cells that will later be incorporated into lateral organs such as leaves. And then we have a centrally positioned rib zone, RZ, which contains dividing cells that will give rise to the internal tissues of the stem. All right, pictured here is a shoot apical meristem in A, and this is divided into these coloured regions on the basis of those zones that we just described on the previous slide. So we've got the central zone, CZ, and two peripheral zones, one on either side, and then the rib zone. And the SAMs also are divided into distinct cell layers. So the outermost cell layer, L1, will give rise to the epidermis. Then we have the subepidermal layer in green, L2 layer, and the inner L3 layer, which will generate all the internal tissues. And that's described here for you. All right, so the architecture of the shoot mainly depends on the production of lateral organs, leaves and branches. And of course, this also plays an important part in establishing that polarity, the basic polarity and growth axes of the plant. And root systems, of course, also increase in complexity by outgrowth of lateral roots. And here's a nice example. The characteristic arrangement of leaves on the shoot is termed phyllotaxy. And there are three basic phyllotaxic patterns that we can see here: alternate, decussate, and spiral, as pictured on this slide. Okay, so they've done studies in both tomato and Arabidopsis, and these studies have shown that leaf primordia are initiated in the shoot apical meristem where auxin accumulates. And this figure illustrates the sites of leaf formation around the shoot apical meristem and how they're related to patterns of auxin transport. So the patterns of auxin movement are actually inferred by localising the pin proteins. So remember those proteins that transport auxin from cell to cell, and these pin proteins are indicated here by P0, P1, P2, P3. And these numbers refer to the age of the leaf primordia, with P1 being the most recently formed, and P0 being the site where the next leaf primordium will actually form. So this newly formed leaf primordium, P1, actually is thought to act as an auxin sink, which prevents initiation of new leaves directly above it on this meristem. So the emergence of that mound where we saw the leaf primordia develop, or a ridge of cells on the side of the shoot apical meristem, represents just one step in the formation of a leaf. So in addition to forming the vascular tissues, development for most leaves features the development of a characteristic planar or flattened form, and this enables an efficient means to intercept light and promote gas exchange, and also enables that highly regulated transpiration process. So leaf geometry can be described by a proximal distal axis, that is running from the base to the tip of the leaf, a lateral axis that runs from edge to edge, and a adaxial abaxial axis, which runs from the upper surface to the lower surface. And of course, you can compare this one to dorsal ventral axis in animals. So the mechanisms by which this type of leaf patterning occurs is still unclear, even though they've identified several important genes that seem to contribute to leaf patterning. So, for example, an interesting one here, the Fantastica mutant, actually fails to establish this leaf patterning, and instead the leaf forms a cylindrical shape, which we'll see on the next slide. All right, so let's look at wild type, so normal leaf patterning here on the left. And according to current models that account for patterning of a leaf, there are interactions between the two tissue layers here that make up the adaxial and abaxial surfaces. And these interactions are required for this normal lateral growth to occur. You can see there and there. And then finally we can see in the mature leaf that laminar shape, the planar shape of the leaf, and we can see distinct cell and tissue types. That is, we can see the adaxial epidermis at the top, and then the palisade mesophyll in the upper portion of the lamina, spongy mesophyll down here. Then we have that lower abaxial epidermis and that centrally positioned vascular bundle there. Okay, what happens in the mutant? The Fantastica mutant. The patterning and differentiation that we just saw has failed. So there's no distinction here in the Fantastica leaf, no distinction between upper and lower epidermis, no lateral growth, no differentiation between spongy and palisade mesophyll cells. And I have an image of that mutant right here for you. There it is. So this is the wild type leaves, and here are the leaves in the Fantastica mutant. Okay, same plant, Antirrhinum. And another interesting mutant here, Fabulosa 1D, although I'm not sure what this one's all about. Okay, so the Fantastica mutant affects leaf polarity. Common mutation could be. It could be. I really need to look that one up, the Fabulosa mutation. But it certainly looks, once again, as if there is a problem with leaf development and leaf polarity. Absolutely, very interesting. And, as we said, they've identified actually a few important genes, but they don't know exactly how they work, how they contribute to leaf patterning. Okay, what do we have next? All right, now let's take a little bit of a closer look at the formation of lateral roots. So the first signs of lateral root development involves divisions of a small number of cells in the pericycle cell layer. This leads to further cell divisions and growth until the new root tip ruptures the endodermal and cortex layers of the parent root. At the same time, a new root apical meristem is established at the tip of the lateral root. And the pattern of lateral root development appears to be influenced, once again, by auxin concentrations, as well as environmental factors such as soil nutrient availability. And this is pictured here on this slide. So these are the 6 stages described for the formation of lateral roots. So we can start with this layer of cells here, which is taken from this little boxed region here. And we can see that some of the cells here are starting to divide just along part of the region here. Further cell division and so on, further cell division until the actual lateral root starts to emerge from the side of that primary root. And then finally, in stage 6, we have all those different cell types and initials, including formation of the quiescent centre and so on. So what's still not known is what actually provides the signal to only some of the cells along here to start dividing. There are only certain pockets of cells that are given those signals. And that's something that's been investigated for many, many years now without any answers. Okay, finally, we're at the end of the lecture. Here we have a very nice overview of the major plant hormones, which have been shown to regulate just about every aspect of plant growth and development. That is if we look at all of the hormones together. So this is the topic, plant hormones is the topic of lecture 13, which we've mostly covered in the prac workshop and investigated in the prac workshop. So what have we looked at? We've looked at the role of ethylene. We've covered ethylene. We looked at ethylene in senescence. We've looked at auxin quite extensively in apical dominance, vascular patterning, cell elongation. We looked at gibberellin, GA, as a plant growth regulator. Although unfortunately our dwarf peas don't seem to be GA deficient. So that particular prac didn't work. So we need to fix that for next year. All right, what else have we looked at? We looked at, yeah, well, I believe, Mill and I believe that those dwarf peas that we've been using, and after speaking to Alistair today, he said he's always had the same problem. Those dwarf peas are not actually deficient in GA. So where that they could be dwarf because they're deficient in genes that are regulated by GA. So any of the hormones act by regulating the expression of downstream, what they call downstream target genes. So they might switch genes on or switch genes off, and so on. So in the dwarf peas that we had, they might have plenty of GA, but it could be one or more of the genes that GA normally acts on that are mutated. So they no longer respond the way they should, and that's why the plants are dwarf. So that means you can apply as much GA as you want, and those plants won't get any taller because of those mutated downstream genes. Anyway, that's our belief, and hopefully hopefully we can actually source GA deficient dwarf plants so that prac will actually work the way that it should work. Anyway, that's our theory. So what else did we look at? We didn't actually look at cytokinin. We just mentioned it briefly in this lecture tonight. We did look at abscisic acid, ABA, in terms of its role in stomatal guard cells and also, of course, influence on potassium levels in guard cells. So we've covered that. So what you still need to go over in that lecture on plant hormones is just cytokinin and the, I think there's a little bit on the brassinosteroids. So I think there's only a few slides between the two of these hormones, so make sure you have a look at them. I don't think jasmonic acid or SA or strigolactones are mentioned. Okay, so that's it for tonight. But before you go, I'm very happy to report there will be a practice exam which I'll load onto Moodle sometime next week, and I'll let you know once that's up. So that's good news. And also, I think there's only one lecture that I did not cover, and that is on nutrients. But the reason I didn't cover it is because it's really quite a simple lecture. It's quite simple, straightforward. So if you listen to that particular lecture, I know you'll be fine. No, there's no. Oh, there is a class, a workshop, a photosynthesis workshop, in-class workshop for anyone that needs help with photosynthesis. Yeah, so that's on next Thursday, but no online class next Thursday. Is that what you were referring to, Laura? The photosynthesis workshop? Yeah. So the photosynthesis workshop, it's in class. It's not in the lab. It's optional. Yes, it's optional. So that's basically designed for students that really struggle with the concepts of photosynthesis, and they need a bit more help with that. So that involves modelling, clay modelling. Is it clay? Play-Doh modelling of the different parts of photosynthesis, and then presenting the model that's constructed in class to other students. So it's quite useful, you know, as I said, for students that really don't get photosynthesis. But once again, that's optional, so it's up to you. That's up to you. No worries. Thank you, Lachlan. And thanks for coming on board tonight. You've got another day of wine chem tomorrow in the lab, haven't you? Yep. Thank you, Laura, also for coming on board tonight. It's going to be a hoot. Oh, it's terrific. Wine chemistry. Great stuff. All right. Thank you, both of you. And I will definitely be in touch via Moodle. Okay. All right, have a great week. Bye.